Hey, so this is the last video for the carbon containing molecules, and this one is just going to focus mainly on, what's the most important thing, which is amino acids. Really, really important. So let's get started. So what do we know about amino acids? Amino acids basically, they have an amino group, they have a carboxyl group, and they have a side chain, and then they have a carbon atom, right? But there are 4 separate families for amino acids, and we know that as, acidic, basic, uncharged, polar, and non-polar. And there are 20 amino acids that kind of account for all of this. So at pH 7, both the amino and the carboxyl groups are ionized, so you need to remember what that pH is. So just remember that for the future. And just like there's glycosidic linkages for carbs and phosphodiester linkages for nucleotides, we have peptide bonds for amino acids that bind to each. So they bind from one carboxyl group to the other. So that's basically what happens. So energy from nucleoside triphosphate hydrolysis. So what does that mean? It basically means is when each amino acid is binded together, a water is produced. Kind of like the same thing as phosphodiester linkages. So proteins, what are proteins? Proteins are long polymers of amino acids that are basically just linked together by these peptide bonds. And proteins are always written with the N terminus towards the left and the C terminus towards the right. That's why it's NCC, NCC, NCC. So one of the main things that we're going to have to learn is how to make these polypeptide chains and all of that. So the best way to learn all of this is just to memorize those amino acids. And yeah. So what other stuff do we have to talk about? Okay. For special amino acids, met, so M is methionine, is always the first amino acid. Just remember that for future. Carbon is usually those covalent disulfide bonds by oxidization and all of that. So now we're pretty much done with that side. So we can move on to the rest of the stuff. So that's pretty much it. So when we move on to different topics and all of that, we really need to get into the basis and understand amino acids and their protein structures. So, we talked about how pH 7 is when the structure of the NCC occurs, and the amino terminus has a pKa of 9.8, so that's when the acids interact with the base. So a lot of protons are acidic and they're protonated. If the pH the solution is below the pKa in a group, that just means that it's protonated. And there's other, terms like glycosylated, that's when we add sugars. Phosphorylated is when we add a phosphate group. So always remember those two terms, glycosylated and phosphorylated. Those just mean we either add sugar to the amino acid, that's when we say it's a glycosylated, or when we add a phosphate group, it's phosphorylated. So what now? So we can't really understand proteins without understanding their basic structure. And basically what structure is, the primary structure is just a string of amino acids that just gets folded. And in this primary structure, the hydrophobic side chains are buried in and the polar side chains stick out. So what does that mean? So when you have that string of amino acid, it's usually folded in aqueous environments. It's not always going to be unfolded. So when they end up being folded in aqueous environments, the hydrophobic core region contains all the non-polar side chains. And then the polar side chains on the outside of the molecule can form hydrogen bonds with water, which is what we need. So those stick out. And what else? Then we have modifications. So modifications to amino acids can change the structure. So it can change the phosphate group, the methyl group, the acetyl group, the sumo group. What other groups? The ubiquitin group. So a lot of this has to do with modifications. So modifications to amino acids can change, substantially change their structure. And we see that with phosphates, methyls, acetos, sumos, and ubiquitins. So keep that in mind in the future. And again, we talked about how covalent bonds are by the sharing of electrons, and ionic hydrogen, van der Waals, hydrophobic, they aren't covalent. So that's basically it for that one. So basically what Stein worked on for her, I think, it was one of her projects that she worked on. She worked on a CRP, and the best way to remember that is it means crap. It doesn't mean crap, but you can use the word crap to remember that it's CRP. And it has 5 subunits, and it kind of forms a donut. And there's different protein structures on each side of it. So these 5 identical subunits, it's one polypeptide chain each, and they bind to one another, and they form a donut. So that's where the donut comes in. And there's 2 sides with different activities, and it functions in immune system defense. So it's good for you to know that, just in case she asks that on the exam. And now we can move on to primary, secondary, tertiary, and quaternary structure of amino acids. So we already talked about primary, but then what the heck is secondary structure? Secondary structure is the alpha helix, and that's the one where you have the spiral staircase. So the amino acids are set up this way. And then you can also have alpha helix that form together, and these alpha helixes are formed together with hydrogen with the backbone and nitrogen and hydrogen of the residue. So what does that mean? So basically for alpha helixes, there's hydrogen bonds between the backbone carbonyl and the nitrogen and hydrogen residues. So that's pretty much all that you need to remember, is that there are hydrogen bonds between the backbone carbonyl. So this whole thing is hydrogen bonds, nothing else. But there are different kinds. There are different kinds of secondary structures. You either have the alpha helix, and then you have beta sheets. And now beta sheets are just they're just parallel. They fold and they form parallel, and then there's hydrogen bonds between each chain. So remember that as well. And with these betas, they can either form, beta sandwiches and beta barrels. So beta sandwiches, it's just beta sheets that sit on top of one another. So they basically pile on top of one another. It's not that much of a big deal. But what else? So that's beta sandwiches. But then there's another thing in secondary structure, which is beta barrels. And what beta barrels is one of the biggest examples of beta barrels is the protein called porin, which makes pores, and that's made by beta barrels. And it has this domain called EGF domain that it folds up the protein that has the D function. So that's basically what it is for the beta barrels. It kind of just looks like a barrel, basically, when it's kind of wrapped around each other all at the same time. So that's basically it for that part. So proteins share similar structures, we know that. So core structure remains the same, but there are some differences to proteins in structure, and these differences come in from the domains. So proteins share pretty much similar structures. So domains kind of mean the sequence of amino acids with particular structure and function. So domains have functions, and these different domains have kind of changed along the line. So through evolution, these more domains have been added to different, organisms to, signify different functions and all of that. So we may add domains over the course of time. So there are some duplications that happen. So, but changes in nucleotides and amino acids, and then with natural selection, that change structure and function somewhat. So always remember that for domains, how important they are. And now we're going to talk about kinase and tyrosine domains, and that's going to be coming up next. Okay.